Okay.
Thank you once again, Matt. So our next speaker lined up for today is Dr. Qiuwei Xu, the Senior Principal Scientist at Merck. He is the Senior Principal Scientist at Merck and leads quantitative NMR operation for metabolomics and vaccine process qualification. The NMR Metabolomics lab is automated for quantitative profiling of endogenous metabolites and assisting investigation of drug toxicity or fermentation bioprocess. The endogenous metabolite identification is facilitated by an establishment of NMR reference library of about 800 endogenous metabolites and software for batch processing of metabolite identification and quantification. He has published numerous papers in methodology and application of quantitative NMR for metabolomics and vaccine research and development. Today, his talk is entitled Examples of Quantitative NMR Analyses in Pharmaceutical Industry. And without further ado, Qiuwei, you can begin sharing your screen and the floor is yours.
Thank you. Thank you, Carrie, for organizing the symposium, and also thank CIL and Krista for inviting me to give a brief talk. So let me share my screen. Let me know if you can see it. Can you see my screen?
Yep, but it is not in presenter mode yet.
Is that okay now?
Yep, you're all set now.
Thank you. So I think first I would say that I really appreciate and I have a long, close interaction with the CIL because we got, isotope labeled chemical small metabolites for trace analysis and also the solvents. And as you can see, I think I always treasure the year-end Christmas cards in my office. You can see typed, I think it's been more than like 10 years. So today my talk is about the role of NMR in pharmaceutical industry, particularly in the case of in the Merck, and I support the drug development and also manufacturing. And for NMR, because of its unique property, and as you see all the talks today for very sophisticated application for biomolecules, but actually the NMR is very unique for its property, quantitative nature and repeatability, and it actually is very useful tools solving lots of issues and the investigation. Here I'm listing a few aspects. One, as you can see, solving the to confirm the structures, either chemical structure or three-dimensional conformation, as also you can see from today's talk. And for NMR, it also can be used for quick identification of chemical components, for example, in the fermentation media, and quantify the chemicals in drugs and in the metabolomics, and also characterize physical properties. So today my talk will be covering following aspects. One is about the polysaccharides, and the second is about fermentation, metabolomics, and also using it for actually physical characterization in support of quantification of chemical components. I will illustrate that. In industry, one thing we always want to achieve is make sure it's a high quality, not just for the drugs, but all the process to support the drugs. So, and same for the NMR. For even quantitative analysis, we require certain criteria of spectral quality and reproducibility. In this example, I show that this is very typical 62 or more than 60 examples based on the groups, and we want to make sure the running through the whole sets of example and the line shape is very consistent. So that's always we want to achieve, be consistent. The second, that in some cases, we not just want to solve the structures in terms of chemical structure, but we want to know the chemical structures relate to the NMR spectrum pattern. In this case, for example, metabolomics, and in order to avoid looking for every single chemical structures, we actually established the 800 endogenous metabolites library. Essentially, we can using software to query all the chemical structure based on NMR peaks, as illustrated here, and we can call up the library to match the the peaks on NMR spectrum. That really saved the times and to give us the chance to query the biological question rather than to solve individual chemical structures. But also we're using different tools. For example, NMR is very good. It's very additive, quantitative in nature to account for individual contribution based on amount of moles in the solutions. And but there is also disadvantages that there is overlapping signals as illustrated, here on the right side or the left side. There are overlapping signals came from individual contributions. So we also explore the opportunity to using the linear deconvolution methods to deconvolute the overlapping components. So illustrated on the low left side is bacterial polysaccharides. For example, in our vaccine, there are 23 different serotypes. And if you overlap all those signals, that's what it looks like in this middle section, either by combining the containers or mix the 23 different mixtures, or we can also simulate using the reference individual standards and then add them together. And you can see they are very similar, even though there is slightly some difference. And using this deconvolution methods, we can actually put all 23 valent polysaccharides and deconvolute to find out individual contributions. And this is the residual from the filling. You can see really nice results. Similarly, this method can be applied to, for example, metabolomics. And we can get a background, for example, the lipoprotein profile based on the diffusion, for example, the profile, and the very sharp small molecules spectrum can be also added onto this background and make this overall, the biological fermentation, for example, or biofruits solution NMR spectrum, and using the convolution, we can get those kind of individual components. So given those, the feature of NMR and the tool we have for quantification, this example is applied for metabolomics approach. And on the left side, we use this quantitative methods. As I mentioned, that we have 800 library endogenous metabolites reference spectrum to identify, all the endogenous metabolites. And in this case, we treated the mytube cells with two different toxicants, which is a toxicity to the mitochondria. One is a rotenol complex 1, and antimycin A is complex 3. And you can see they change the different pattern from the glucose, glutamine. Just like Dr. Merritt was talking about the mitochondrial toxicity, you will see the pattern. For example, the lactate will increase in the red. You can see those in the presence of the mitochondrial toxicants, in this case, rotenol, red, and antimycin, green, compared to the vehicle control. So this just illustrates all the metabolites we can list. Some of those representative here, and we can look into the metabolites to understand what kind of a toxicity to what kind of a detailed mitochondrial complex inhibitions. On the right side, we're using this metabolics approach. This is kidney toxicity with the gentamicin and cisplatin. And we're using this approach, quantify the urine metabolites. And also we're looking into the transcription pattern change in the kidney tissues. And we can construct the almost like the biochemical process of the toxicity, how that affects the membrane, transporters, and how that affects the nutrients recycling, because kidney actually is a very unique organ. It not just dispose the waste, but actually recycle nutrients like glucose. And when they have impaired, damaged kidney, and that will lead to, for example, leaking of the glucose into the urine. So that's help using this NMR approach in combination with the transcription, we can construct those biochemical toxicity. This slides illustrate using quantitative NMR to looking biochemical pathway, very similar as the speaker talking, I think, today. and also Professor Merritt were talking about, we're using actually to understand in Hep2 cell, it's immortal cell lines, it's a cancer cell lines. And we're looking into the C13 labeled galactose versus C13 labeled glucose to see what's the change of metabolism. The purpose of this one actually is to support our assay to look in mitochondrial toxicity and to try to understand what's the difference of these two nutrients. But what we found was interesting was that if you have a glucose, just like any cancer cell, they will metabolize the glucose undergoing anaerobic glycolysis, which means that most of the products did not enter into the TCA cycle, but just purely go to the glycolysis, end up into lactate. But if we feed the galactose, what we found that is that in the cells, we didn't see converted into glucose, nothing to convert it into even the lactic acid. And we only see the intermediate metabolites from the galactose feeding, for example, into the galactonic acid is essentially oxidized into carboxylic acid. And of course, we see also intermediate metabolites ahead of it in the galactose phosphate, but nothing converted into glucose or lactate. So essentially, this illustrates that in the cancer cells, if you feed the galactose, and actually the nutrients cannot be facilitated into nutrients of glucose and into lactate or TCA cycle. And this is a very unique feature of cancer cells. And people actually thought about, deprive the cells with a glucose, maybe feed the galactose. Maybe this cancer cell cannot grow and stop. But in practical sense, it's really difficult because other normal cells still need the glucose for metabolism. Here I'm switching to another aspect using quantitative NMR for vaccine products release, for example. And in this case, we also monitor the process of making conjugate the vaccine. In this case, it's a bacterial polysaccharides, and essentially it has to be linked to the protein to make a conjugated vaccine to give immune boost effects. And this is a pediatric vaccine, and a Haemophilus B bacterial polysaccharides conjugated with meningococcal proteins. So we use the NMR. First of all, we quantify in each production or processing medias how much this bacterial polysaccharide contains. We're using quantitative NMR approach, and also during the each individual processing, we call the activation conjugation process. Actually, we characterize it using the NMR to see how much this the Haemophilus B polysaccharides is conjugated with a linker and with the activator bromoacetic groups, and also we characterize how much activity bromine groups remain, because sometimes when we make manufacturing them in the media may not be used instantaneously, maybe holding on for a certain time, and actually bromine were lost. And we're using the NMR approach to characterize how much bromine actually remained in the intermediate production steps. So this is the example using the quantitative NMR for our product process control and the product release. The last but not least are also illustrated using the NMR for characterizing hydrodynamic sizes, but that's not the purpose for characterizing the polysaccharide for vaccine. But actually, we have a question about how to quantify the C polysaccharide from the bacterial polysaccharide. So let me just give you a little bit of illustration what this is. So in the bacterial polysaccharide, and there are different codes, just like different codes wrapped around the bacterial polysaccharide. And the capital polysaccharide, that's what we used for vaccine. And that's what elicited the antibody in the human and to protect against the bacterial infection. But it's a very complicated structure, actually, unlike small molecular drugs. actually inside there is a layer called the C polysaccharide, and it's always tethered to the capsule polysaccharides. And so during the manufacturing, and that C polysaccharide, even though it's not immunogen, but it's left behind, and it will actually compete for the chemical reagents for making conjugated vaccine for capsule polysaccharides. This is the NMR spectrum illustration of a C polysaccharide, and the prominent signal is the coding signals. And for our capsule polysaccharide, we don't have the coding signal. So that's very unique for this C polysaccharides. This is the bottom one trace is the pneumopolysaccharides, any one of those 23, for example, and the polysaccharides. And you can see it's all dominated by the capsule polysaccharides, but there is a trace around the coding inside. The question of how much you see polysaccharide, that's pretty straightforward. You can integrate it, and you can calculate with respect to bacterial polysaccharide. But the question coming with that, how much actually is tethered to the capsule polysaccharide, how much is actually free in the solution, even though they look very similar on the NMR spectrum. So the way we try to separate these two using actually diffusion NMR, and we're using two not say bounding mode, right? Essentially two population mode approach. One is tethered to the bacterial polysaccharide, another is free. So we use this mode to deconvolute the diffusion NMR curves, and essentially we can get how much is tethered to the bacterial polysaccharide outside the capsule polysaccharides, and the remaining will be just a free polysaccharide. But in addition to those quantitative analysis, of course, we come up with other physical parameters, which in terms of the hydrodynamic radius, diffusion constant, that actually is useful to understand those kind of process, the polysaccharide, the size, their physical attributes. But I think for a particular application, we actually use just to get how much is bound, how much is free. Although there's another technique, for example, size exclusion chromatography, and the measurements for using combination with some fluorescent labeled detection. But I think the drawback is that NMR advantage compared to drawback of those size exclusion chromatography is that NMR is much easy. It doesn't need a manipulative samples, doesn't need to tag any fluorescence tag, and it can go to the solution and do the measurements and just do the mathematical fitting, and here is quick results. So, and also the ambiguities in the size exclusion chromatography, you have to cut which belong to the free, which belong to bound. Sometimes is well resolved, sometimes have overlap. So definitely they show the advantage of using NMR for this kind of applications. So yeah.
Oh, okay. Yeah, if you're wrapping up, that's great.
Yeah. So I as I say, this is NMR is not only for structure. Actually, quantitative NMR is very, very useful in pharmaceutical industry for fermentation, biological research. And I think thanks for the NMR instrumentation, now it's very stable. You can do the automatic shimming. It's very consistent with the line shape. And of course, as I say, for the whole operation, not just the NMR instrument, there are other things to consider. For example, the solvent, can be, quantitative, solvent need to quality control the reference standards, and also need a good software for chemical identification, and also for much batch process quantitative deconvolution. Thank you for your patience. And here's just my colleagues, and thanks for CIL for the support over the years. And these are my colleagues. And for sake of time, I think I'll pause.
Wonderful. Thank you so much for this excellent presentation, Qiuwei. Unfortunately, due to the timing, we're going to have to move on to our next speaker. We do have a few questions in the Q&A box, so I will go ahead and I'll post those into the chat. If you would be able to address them there, that would be greatly appreciated.
Okay, thank you.
And if you could stop sharing your screen, that would be great. Perfect. Thank you.